I'm Sarah Pagan and I'm the lab manager here in the Human DC lab at Newcastle University. I'm funded 1 day a week by Bright Red to manage the lab and also be involved in public and patient engagement for the charity as well. So today I'm going to show you our new quantitative real-time PCR machine that was bought with funds from Bright Red to help us with our research here at Newcastle University. It's a Quant Studio 3 PCR machine by Applied Biosystems, and it's a quantitative PCR machine, which basically means in real terms that it measures the amount of gene expression in genes in our DNA and tells us how much of that gene is in those cells, and we can then compare that to the expression of genes in the healthy control samples. And then that amount of gene expression can tell us what might be going on with those patients and how we might use that to find out more information about their blood cancer. So the real-time PCR machine reads the samples in this 96-well plate, so we need to set up the plate before we carry out the reaction in the machine. So what we need to do is we add our patient DNA samples and our normal healthy control DNA samples to the plate first, as it's the DNA from the patient cells that have the genes in them that we're looking at to see what the different gene expression is. So all of our cells in our body have DNA in them, apart from our red cells, and when we extract the DNA from those cells, we can measure how much is there, and then once we know how much is there, we work out how much we need to put into the reaction plate for the PCR reaction. So once we've added our samples to the plate, we then need to add in the assay for the genes of interest that we're looking at. So we can look at anywhere between 1 gene and 100 genes at any one time. And we have 1 gene per well, and we make up a master mix with the primers for the gene that we're interested in looking in, which actually attaches to the piece of DNA and then amplifies it so that it can be read by the machine. So we then add our assay master mix to the plate as well. And when the real-time PCR machine carries out the real-time PCR, what it does is it heats the plate up. Inside the machine there's a metal plate you'll see in a minute. We sit this plate in it, and it cycles through various temperatures, and that amplifies the DNA in the area of interest of the gene we want to look at, and then we get a readout from the machine to tell us what's going on. Before we can put the plate in the machine, we just need to seal it with a lid, and this is an optical lid, which means that the machine can read through the lid and see the colour change reaction in the wells. So to load the machine, we just press the eject button, and if you see, there's a little 96 well heated plate in here, which heats up very quickly and cycles between the different temperatures that we want to look at, and we sit the plate in there. We had an older version of this machine before Bright Red purchased the Quant Studio 3 for us, and the run times were much longer. It was a much more cumbersome machine and took a lot longer to set up and a lot longer to run the samples. So now we have the newer model. It's helped us with our research, saves us time, and we get better data from it as well. So after we've loaded the plate into the instrument, we just need to set up the run on the computer to tell it what we want to look at. So we just tell it what we're running, what size the plate is and what size the reaction is. So we've got a 10 microliter reaction today, and this shows you here the PCR cycles. There's 40 cycles of 3 temperatures, and that's what's going to amplify our genes of interest to tell us what we're looking at today. And then we just have to tell it what we're running in the wells. So we've got 2 genes today that we're looking at, and we just have to tell it that we're running 2 different genes for these 16 samples here, some healthy control samples and some patient samples, and then we go into the next screen and we start the run. I'll show you an example of some of the data that we get off of the quantitative real-time PCR machine. So this is just the plot that we get back at the end of the run that shows us over the 40 cycles of the PCR how the amount of the gene increases exponentially and then reaches a plateau. So the machine measures this increase in fluorescence as the cycles go along, and then these samples here have a higher amount of the genes expressed, and these have a lower amount of the gene expressed because they take longer over the 40 cycles to reach a certain threshold to show us how much of that gene is expressed in those samples. So we take our DNA from our patient blood samples and compare it to the healthy controls, and then this can tell us information about what's going on in those patient cells and help have more information about what's going on with their disease.